This is Project REDS. It stands for Rapid Ebola Detection Strips.
Okay, so to start off, we're going to give a little background on the Ebola virus. Next slide, please. It was first discovered in 1976 in the Democratic Republic of the Congo near the Ebola River, hence the name Ebola virus.
Since then, there's been many outbreaks. This one has been the worst one in terms of casualties.
And what's unique, before you switch slides, what's unique about this outbreak is that typically Ebola sticks around in East Africa. And the thing is now it's gone over to West Africa, where individuals have never really experienced this virus before. And so this is the case for us as well as Westerners. And the thing is, this is particularly devastating for the public that hasn't been exposed to a virus, and that's when you start seeing upwards of 90% fatality versus 50%, which is what you'll be seeing in mortality in East Africa. Next slide, please. And the Ebola virus disease is the disease that Ebola virus causes. Can you click it again? Okay, go back. There's supposed to be text there. In any case, the Ebola virus, there are 5 different strains, all but one cause disease. And the viruses basically are genome holders. And some are made of DNA and some are made of RNA. The Ebola virus is an RNA virus. Next slide, please. And what's typical of viruses is that there's typically kind of like a niche in which they kind of, they've adapted with their host. And so for the Ebola virus, that's the bat. Now they can also go into different species. And so they can cross species into primates and also into humans. And so when it crosses species, especially when a host has never experienced the virus before, like I mentioned before, this is when it causes a lot of disease. Now eventually we can co-evolve and be okay with it, but we want to avoid the casualties in the meantime. Next slide, please.
Okay, so Ebola is transmitted through contact only. Now contact can occur either human-to-human contact, or like Dr. Liepkalns said, human contact to contaminated fluid from, say, bats or apes who have the virus in their blood. So one of the main ways that people in Africa contract this disease is through hunting bushmeat. And that's not from the consumption of the bushmeat itself, but from the actual hunt. So say hunters have open orifices or lacerations on them, and they go through and they're hunting fruit bats, and they get contaminated guano or ape blood in those orifices. Then they contract the disease, bring it back to their village, and then spread it to everyone who's there.
The ways it doesn't spread is through mosquitoes or insects, air, water, or foods bought in the U.S. And Brian mentioned the bushmeat trade. Something I recommend to watch is a Vice News documentary. It's very compelling, and it shows how they prepare it and how they hunt bats and primates.
So treatment. Currently, Ebola virus is only, the symptoms are the only thing that can be treated. You can't treat the virus itself. So this is very difficult to do in West Africa due to the lack of modern medical resources. However, there are current antivirals that are in development, although none of the FDA hasn't approved any of them yet, although they're scientifically in studies showing a lot of promise. In addition, there are 2 promising vaccines that are in development that are actually scheduled to come out this July. So we're actually looking forward to that.
Now, signs and symptoms, since there's really no very specific, well, there is one specific way to really tell if you have contracted the disease, but there are some limitations. And so if you're traveling, for example, because really we don't want the virus to spread, one way to tell whether or not someone is, what we're using today to tell whether or not someone has contracted the disease, really is looking at fever, headache, vomiting, and all of these are really common with other diseases that are not fatal. I mean, what if you just have a headache because you didn't eat enough? What if you have a fever because you have the flu? What if you have something else that really doesn't cause? So a lot of times, a lot of travelers will be stopped coming from Africa thinking, well, maybe this person's contracted the disease. They're put into quarantine for up to 21 days, which is something that no one really wants to experience. And so that's one of the major limitations of what's going on today. And so, in addition, Yes, that's right. And so another thing that's happening with the virus is that up to now, I mean, the Ebola virus has been present in Africa for a long time. Now it's come and gone from different villages because it really killed so quickly. You would die within a week. So it didn't go very far. The thing is now the virus has mutated, where the incubation period has increased, meaning from when you actually get the virus to when you actually show symptoms of the disease. It went up to 21 days. Now this means that you can travel thinking that you are completely fine with no symptoms, even though you're carrying the virus. Basically the virus can hide within you until you go into another area, and then you can start developing symptoms, and that's when you actually start shedding virus. When you have that fever, when you have that headache, that's when you start actually shedding virus, and you can be contaminated. You can contaminate other people.
So where this is relative is to traveling. People flying from Sierra Leone, Guinea, and Liberia are on a warning level 3 from the CDC, meaning avoid any non-essential travel. For what they're doing is they're screening all the passengers from those flights and making sure they don't show those symptoms. If they do, they're quarantined for the incubation period and then released after that. And that's one place we want to distribute our red strips as terminals to provide a more holistic and effective screen.
Now, the Ebola virus can be present in all kinds of different fluids, including the blood, saliva, urine, tears, semen. And so a lot of these, as soon as you show symptoms, like I mentioned earlier, that's when you start seeing the presence of virus. Now, if we were to detect the actual level of virus, which is how it's diagnosed today, typically blood is what's used. Now, saliva could be a potential way of actually seeing viral loads. What's interesting too is that even after months of being, after the exposure, you can still see virus in the semen. Now today, and during laboratory testing, there's something called real-time PCR. And so basically what it does is it looks at the genetic material of the virus, basically the guts of the virus. You make a lot of copies so you can physically see it. And so if you can see that there are genetic material within your blood, that's when you say, okay, this person has the virus within them, this person is Ebola positive. The thing is though, this is a very big, bulky machine. It takes training to actually use, and also it takes time.
So Justine mentioned that this shows like the blueprints, you know, it amplifies the RNA within a virus. And I spent a year working with, a summer, doing PCR, running it. It took me an entire summer just to learn that process. It's very cumbersome. The reagents are expensive. The machine I use cost over $100,000. And these are reasons why it's not very conducive to the climate in West Africa.
So if we already have a way to detect Ebola in the form of PCR, then why use strips? Well, like Rostam said, it's much more affordable. The initial cost of buying a PCR machine is anywhere between $10,000 to a couple of $100,000. More portable in Africa, PCR are heavy and they require constant electrical output. So with our strips, no electricity required, they're very light, so you don't need any of that, so they're much more conducive to being used in, say, an Ebola outbreak setting. They're faster. PCR needs a couple of hours to run, sometimes overnight, where these strips can just be used in under 30 minutes. And they're more user-friendly. Like Rostam said, he spent an entire summer in a lab under trained professionals learning how to do PCR, which is very complex. I mean, I can't even do it yet. So these strips, all you have to do is read the instructions on the packaging, and you can just go to work right away.
Basically, all it is just a piece of paper that'll change color. You put a drop of blood or saliva or any type of bodily fluid, in which case the virus could be present in. You put it on there, and just like a pH strip that you would check your pH of your pool, it'll change color, and that's a pretty immediate thing. And
as far as why the time is important, you go with the PCR and you test a whole village, it takes a couple of days before you get the results back. And then by that time, those people can be dispersed from rural areas, take a taxi. This actually happened. You know, a person with a rural area took a taxi into a city of 12 million people, and then suddenly you lose all track, and the people they come in contact with, the big part is contact tracing, and you can't do that if it's spreading like wildfire. So once we had the strip idea, we needed money. So we talked to an MBA student, and we decided to go with crowdfunding, mainly because it would raise private capital, Unlike grants, it would give us more flexibility with our project. Our goal was to get 14,500 in a month, and we surpassed that in less than 3 weeks. A large part due to our Emory community, as well as my high school and our high schools, and some people who are in this room today. Thank you. And then once we had the idea and the money, we knew we had to protect it. So we filed our temporary provisional last October, and we're looking to get our full utility by the end of the year.
So this is our lab at Emory University in the biology department. It was generously donated to us by the head of the biology department at Emory University, but it wasn't so easy as it was just handed to us. We tried to get the lab through traditional university channels, and we just kept getting stonewalled at every turn. So Rostam and I, after seeing this, we took a step back and we looked at our network. We're both students in the biology department at Emory. Dr. Liepkalns is faculty in the biology department at Emory. So we went to our professor and head of biology department, told them our situation, and they graciously donated an entire lab space to us, which we are very fortunate.
So this project was anything but traditional. What's typical is that you get hired as a faculty who is supposed to lead research with students, and the thing is, this instead came from 2 students, and then we went to the biology department, who really, who gave us this space because they had faith in our project.
So this is probably my least favorite part of the process, but it was really educational for me because I have trouble understanding why some people do things at times. And I guess a lot of times you have to realize that norms aren't always there because they're right, they're there because they're popular, and a lot of times it's actually an ineffectual way to do that. However, when addressing that and questioning authority, it's important to be polite, respectful, and productive.
I hear laughs.
All right, so Rostam and I, there's actually a really funny story between how we met. So we're both in Raoul Hall at Emory University. It's a freshman dorm that's based on social entrepreneurship. So in order to be placed in this dorm, you have to show some aptitude towards social entrepreneurship. And one night, about a week before school started, I was just walking down the hall, minding my own business, and I hear an unfamiliar voice yell out behind me.
So I had saw Brian walk by, and I asked him, did you wrestle weight class 138 or 45? And he wasn't wearing anything that had wrestling on it, but I could just tell because I had a great faculty and staff, my coaches in high school, who's right there. Shout out Coach Kip. And so we decided we were wrestling partners before we were lab partners, and that is something I really want to pause and reflect on. And that's, whenever you do a venture, there's gonna be failures, there's gonna be successes, but either way, you're gonna enjoy it with people who have the same mindset and that people you just like being with. So once we got that going, we looked to our community because this project, a lot of the momentum came from our local community. As you see, that's a postcard sent out to 50,000 high schools nationwide, and that's where we were first working the process out. Our hall that Brian mentioned is literally right across from the CDC, so we can see it. So it was really cool to draw on the board and to be like, Ebola is like right across the street. And during this process, it came from everyone. Emory students were like rallied behind us, really supportive. Their parents got like double as excited, all our advertising, our website development, our incorporating the company, all was done pro bono from parents. And then my favorite part of it was local kids. The local Montessori school invited Brian and I to go speak to 7 and 8-year-olds about Ebola. And so driving over there, had my slides on like case mortality, internal hemorrhaging, and I was like, this might not be appropriate for 8-year-olds. So I changed it to like spaghetti, meatballs, and it was actually really fun. I enjoyed doing it, and I learned that you really don't know what you're talking about in science unless you can explain to an 8-year-old. People use these big words to like scare you, like exoskeleton. That's an Iron Man suit. So you just gotta like put it in the right terms. And that was a big part for us. And they said the kids loved it so much. Can we raise money for y'all? We're gonna do a little bake sale, and that was the kids' idea. We're like, okay, chocolates and cookies will make like 20, 30 bucks. Awesome. And they came back next week with like $300. And so shout out to Montessori for that, and we were super grateful for that.
All right. So when we came up with this idea, we understood, since it was our first venture, that there was a few steps that we had to follow. The first was idea generation and just the solidification of our idea. So we couldn't just have it floating around willy-nilly in our head. We had to get it down on paper because we're college students as well. We're full-time students. We have other obligations. So we can only allot so much time to this project. So we needed to make sure that the time that we did allot was spent productively. Next was finding a team. Rostam and I feel fairly confident in a lot of scientific and business ventures, but we knew we needed some credibility in the form of Dr. Liepkalns and an MBA student who is working with us on the business side. And then we needed funding. You know, money makes the world go round. We can't buy our lab equipment without money. And what goes hand in hand with that is publicity. In order to people to want to donate money to us, we need to show them that we're legitimate, that we're trying to do something good for the world. So that also helps a lot. Then, like I said, networking. Networking helps a lot. So when we tried to go through the traditional channels, like I said earlier, with the lab, they weren't working out for us. So instead of having to spend months and months in that red tape, we were able to go to our network and get a lab within 2 days. So that was amazing. And then finally is execution, which is the stage that we're at now. So it's all about time management and focus. Like I said earlier, you need to manage your time wisely and then just bear down and get in that focus so you can get as much done as possible as quickly as possible.
As far as the setbacks, it was really the same thing, just from different mouths. It was things like it takes time, it's part of the process, that's just protocol. Basically, the same politics, bureaucracy we talked about earlier, the red tape. And what we learned about that is, unfortunately, that's prevalent in many large institutions, but we shouldn't accept that, and we should approach it, though, in a polite and productive way. So, as college freshmen, we obviously have to make the transition from high school to college and like be a full-time student and all that cool stuff. So it was, at what he talked about, time management, that was a big part of it, like managing this project, also with our academics. At the same time, it gave us a unique advantage because college is a place that you have so many resources that can help you to succeed, but just as importantly, you have a safety net that can help you rebound from small failures. And then the cool thing about Emory is the undergrad school is right next to the med school and the business school, so we could just go there and talk to professors or business experts about viruses and enterprise at the same time. And then being at Emory during the time of Ebola, it was kind of like right place, right time. So what was our niche? Unfortunately, Ebola wasn't a big deal to the U.S. until it came to the U.S. So that's why, like, why is PCR necessary, or why is REDS necessary if PCR is already accurate? And the question is, West Africa doesn't have the resources that we have. So having something that's portable, affordable, and easy to train people to use is vital. And of the most important things, Mr. Milton talked about this earlier, you have to know your why. That's just a fancy way of saying you have to find something that really pisses you off. For him, it was, you know, what he explained. For us, for me, it was, you know, why is it that we only care about something until it comes to our doorstep? I mean, it was discovered in 1976. So, like, one of my role models, Dr. Paul Farmer, said the only real nation is humanity, and I strongly believe that. And so to be able to create something that could benefit that was my why.
On the same wavelength as that, I'm studying, and the whole reason I'm at Emory is to become a physician so I can help people, and to have the opportunity where I could create something that could help people now and not have to wait, you know, 8 or 10 years until I'm through with med school and undergrad education was my why I want to invest so much of my personal time and money into this project. So then, another lesson is that we can never be intimidated by authority. It's good to question authority if you question authority in the right way. Do it in a productive way, do it in a kind way, do it in a respectful way, because sometimes authority is there for a reason, so we had to learn that. And also, we had to learn that your ego is not your friend. So in the beginning, Rostam and I felt like we could, you know, take the bull by the horns all by ourselves, but we realized soon that that was not the case, and it was okay to ask people, and it was okay to not know something.
Yeah, and there's, sorry, so another thing that I've learned as well is, the thing is, so I've been doing research for a while now, and a lot of times you do really a lot of work for very little payoff. Maybe you'll get a paper, maybe not. And so this was a project that really had a concrete solution at the end of the tunnel. Also, all of us have our own careers going on. I mean, Brian wants to go to medical school, and Rostam really wants to take a social entrepreneurship seems like a career choice. And I want to be an educator. So when I was initially approached, it was actually a friend of mine that had worked with Rostam over the summer in a lab, said they're 2 students, they got a project and they need a mentor. So immediately I said yes. And so we met at a Starbucks, and then after that we did an Indiegogo where we raised funds. And so this project, that's what really kept me going, because even though this project right now, we have a lab. I mean, the fact that we have a lab after a few months is actually a really big deal. And then if we can make it to completion, even better, we can really make our mark. And at the same time, it can also help our careers go forward.
So other things was be able to hear no. Brian talked about networking. We have to ask so many times, big ask, small ask, but a lot of times we hear no. And at first it's discouraging, but what we learned is those no's shape you just as much as the yeses. Because if you don't ask, you have 1,000 options. When you find out that you ask and get all those no's, you can actually choose which resources are available to you and then plan accordingly. And besides that, comes with that is the fear of failure. Entrepreneurship just an up and down wave, and that's why the why is important, because it's something that anchor you to your vision.
Because I've seen a lot of times people in the lab where they'll design experiments over and over again, never actually get to start it, just because they're afraid of the failure. They're afraid of actually having the experiment fail. But Brian and Rostam have definitely shown that they're not afraid of it. If I've had phone calls sometimes like really late at night, like, hey, I talked to so-and-so, and that didn't work, so tomorrow we're gonna do this. So always, and they're always thinking about it, always pushing it forward.
So with those ups and downs came something I didn't foresee. For me, it was personal health. When you get that stress, that anxiety, the first thing we cut is like exercise, things that are important for us mentally. And in person, we have a proverb that says, a healthy mind dwells in a healthy body. And that really struck home to me through this project, because I realized if you're not at your best, you're not going to perform at your best. I thought like health was something for like really old people, for like 30-year-olds. So I guess it's not.
So another thing that we learned was the importance of community. Like Rostam said before, Emory has been phenomenal in all of its support, not just the faculty and staff, but our fellow students, whether it was having their parents help us out through their resources or their connections, or just being there for us, when we're up late in the study lounges on our 4th cup of Dunkin' Donuts. So that was also really critical to our success. Again, another thing we learned in wrestling is actually to love the grind. Not only love the successes of your work, but love learning from the little failures too. And again, to capitalize on an opportunity. Rostam and I, when we came up with this idea, it was our first day of college in biology class, intro biology class. So we saw what our teacher showed us, how the Ebola epidemic was hurting so many people in Africa and possibly in the West during that time. So we saw that, look, we can catch this wave. We're at the right place, at the right time, with the right resources. If no one else is going to do something, we need to do something.
And on the same line of thought, Brian got published in a peer-reviewed journal on disaster medicine. Dr. Liepkalns got our first primary investigation, or PI position, and we all got to get a foothold for our careers, because we were at the perfect place at the perfect time, and we weren't experts in infectious disease, she was, but we were gonna be, because it fell into our lap, and when something like that falls in your lap, there's no excuse to not take it.
Because really the first day they were in class, their professors gave them an assignment based on the 2 patients that were at Emory Hospital. I don't know if you heard about it in the news. And they actually went and saw the samples walking from the hospital over to the CDC. So there was a wonderful opportunity that really landed at their doorstep. And so they really took advantage of the resources around them. Rostam and Brian went to talk to a PI in their lab, and also talked to someone that I was connected with, and so that's how, and we really operate as a really good team as well. We're really open with each other, and if nothing else, I mean, this is a wonderful learning experience, and hopefully we'll see it through to the end.
Thank you very much. Are there any questions?
time for one or two real quick questions.
What is it, or we're? Well, currently we have the lab, and we just received the green light for ordering samples to actually start the test. The thing is with this test is it's actually really commonly used. It's actually how the pregnancy test works. So it's been done plenty of times, but it's never been done with Ebola because it's been ignored up until now.
So what's unique about the process that makes it work for Ebola as opposed to other applications?
Do you want to take it?
So the blend of reagents we use, right now we're still in patenting, so we can't say that, but they only target Ebola, and it's like it captures the skin of the virus.
Yeah, it's very specific. So these tests are typically, they use a little molecule that's very specific to whatever it looks at.
Is the virus evolving?
All viruses evolve. That's just like we do, actually. Basically, all it is just a mutation. And since viruses have very few genes, one mutation for them can make a very big difference. Also, I mentioned genetic material. So I don't know if you've heard of RNA versus DNA. And so there's viruses that are DNA viruses, and there are viruses that are RNA viruses. And so RNA is particularly unstable and mutates a lot, and Ebola is one of those. But the good thing, though, is those things don't really live too long outside of the body.
Part of that evolution was the incubation period, extending up to 21 days, which makes it much more deadly.
So viruses and us, we're always co-evolving. And eventually, Ebola, I'm sure, because we get sick with viruses right now, but we don't even feel the disease. I don't know if you've heard of adenovirus. It's usually gene therapy. We can get sick, we can get infected with that, and it does not cause disease at all in us. HIV, of course, causes disease in us, but doesn't cause disease in primates. Flu doesn't cause disease in us, but not in birds. And so for Ebola, that's the bat. The bat can have Ebola. They don't get sick. But it's when it causes species to a species that's never experienced it before, or at least not for very long, that's when it causes disease.
Is your product a product that you could see extending into other epidemics like AIDS outbreaks and all the other things that are going on in Western?
Yeah, so we've already been contacted by some medical companies, especially for regards to herpes, but our focus right now is Ebola until the vaccines come out or something like that. Thank you very much.
Thank you.
Thank you.